Hi everyone, my name is Carlos Rosas and today I'll be interviewing Kenneth Siefken, a 20-year-old biomedical engineering student at Purdue University. Kenneth is recognized as one of the top students in his major and he's consistently made the dean's list. He's currently conducting advanced research on calcium signaling in plants, focusing on building a one-dimensional model to stimulate calcium diffusion between plant cells. We are here to learn more about that work and what it means for science and real-world applications. Kenneth, thank you so much for your time today.
Nice to be here.
Okay, let's start easy. How is everything going this semester? Busy schedule, I mean.
Yeah, lots of work, especially research, taking me a lot of time. Classes as well, but the bulk of my work is right now research, so that's what takes up most of my daily life.
Okay, nice. Kenneth, what first got you interested in biomedical engineering, and specifically, how did you get involved in this project?
Well, since I was in high school, I've always liked math, science, and as well as also like medicine and biology. I never wanted to be a doctor, but I did like how to find solutions to biological problems. Thus, I didn't really have that passion to be in a hospital and actually perform surgeries. So I tried to find an intersection between that and math and engineering and science. So I found that biomedical engineering was going to be the best option. Then when I was already at Purdue in my sophomore year, I took a BME 202 class. It's thermodynamics and biological systems. And the professor, Dr. Pinar, I was really interested in the research she was doing in the department. So I reached out to her, I sent her a couple of emails, I met with her, did some interviews, and then she told me that she would like me for me to be in her lab. So I started doing research last semester. So on this year's on September, I'm gonna complete 1 year doing research with her.
Oh, nice. Sounds good. For those who don't know about the topic, let's start asking about what is calcium signaling and why is it important in plants?
So Ca2 plus is the calcium ion. That's a second messenger in plants.
Mm-hmm.
So when plants experience threats such as pathogens, it's important for them to be able to communicate between each other. So calcium is a second messenger. So what that means is that plants can send calcium ions among them to communicate with themselves. So it's basically a communication method that would allow a cell to communicate threats and trigger warnings to other cells. So that's the importance. If there's not calcium signaling, plants wouldn't be able to communicate with themselves. The cells would be isolated and they wouldn't form a whole organism that is intercommunicated. So that's the main importance. As well, plants have something called plasmodesmata. There are little holes in the cell wall. Through there is that depending on the permeability, calcium ions can be sent among cells. So if that doesn't exist, there's no communication. If there's no communication, the organism wouldn't work because they wouldn't be connected. So that's basically the importance of calcium in plants.
It's interesting how plants can have different kind of nutrients and kind of calcium in this case. Can you describe me the motivation behind your research on calcium signaling in plant cells? What inspired you to focus on one-dimensional model for simulating calcium diffusion?
So it's always interesting to be able to analyze organisms other than humans. Plants are one of the are more basic than humans. They're not that complex. So being able to study them and perform experiments on plants is first more ethical and will allow us to know how other organisms communicate between themselves and react to pathogens. If you're able to understand how more basic organisms communicate among themselves to target or inform about a threat, that way after getting that understanding we can move toward more complex organisms and eventually reach humans. And if we're able to understand how human cells communicate between them more efficiently and we're able to get that deeper understanding, we will be able to get human applications and improve our daily lives. But in order to do that, we first need to get to the more simple organisms such as plants. And after we understand them, we can continually progress to mammals and eventually to humans. Why do I do a one-dimensional model? A 1D model is basically, it only moves left or right, it doesn't move diagonally or up or down. So that's very simple to play with. I can vary parameters and I can observe how diffusion occurs in one axis. That makes my research way easier, and I can see how the effects in changing different parameters such as permeability or source strength will affect other cells to the left or to the right. That way I can see more rapidly results. But in the future, I would like to incorporate a more hybrid model, maybe a two-dimensional model, that will be able to perhaps perform a grid of cells and be able to analyze cells in a grid shape and not only linearly. But for now, a one-dimensional computational model is easier to code in Python, and it's easier to understand and to gather results from it for some preliminary research.
Okay, you mentioned something about the different kind of models you want to use and you are using right now. So in simple terms, what are the main objectives of this model?
So right now I'm using this one-dimensional model to observe how that change in plasmodesmata permeability, which is called beta in my code, and activation strength called sigma, would affect the diffusion I'm implant. So for example, I can vary beta to be 0, 10, 100, 1000, sigma same way, be 0, 0.5, 1, 2, 3, and see with those variations, be able to create plots, chemographs, to see, for example, how calcium concentration vary among cells against distance with those variation parameters, or to be able to observe a heat map and see the diffusion between cell wall when I vary the permeability of plasmodesmata. So that's why I'm using this model now. I'm trying to understand the role of plasmodesmata more deeply and see how, by changing plasmodesmata and source strength, I am able to observe the changes in diffusion, the changes in calcium concentration, the changes in reach, and the changes in the firing times of the channels.
Very interesting. Okay, what biological or chemical assumptions did you have to make to construct this model?
So first, it's important to assume that the pathogen that I'm using is found in the wild, so it's a natural pathogen, it's not an artificial pathogen. As well, the cells that I'm using are cells from a plant called Arabidopsis thaliana. It's a plant found in the wild, so I have to make sure that the model that I'm building, the cells that I'm simulating, would comply with the ones found biologically in order for them to be applied. So those assumptions that I gotta make for the model, I gotta assume that what I'm building is very similar, it is 90% similar to Arabidopsis thaliana cotyledon pavement cells that were the ones that we're using right now. So basically those are the assumptions that I'm making, but in the future, most likely I would need to add more agents, more parameters to be able to apply other types of assumptions, such as external factors, such as air, such as perhaps insects or bacteria, and apply that to the model, but that would be future research. So right now, I'm assuming that there's not external factors affecting the cells.
Okay, nice. What kind of data or theoretical framework are you using to stimulate calcium movement?
So for calcium stimulation, I'm using first a source. As I told before, it's a one-dimensional model, so it starts in the middle. My model is 700 micrometers long, so there's a source at 350 micrometers, and that source would fire calcium, fire Ca2 plus ions, and then that's what would spread to the other cells. So each cell is 100 micrometers, so the model has 7 cells in total. If there's no plasmodesmata permeability, then calcium wouldn't leave the first cell because plasmodesmata is 0, so calcium would be stuck there. If plasmodesmata is increased, then calcium would diffuse to neighboring cells, and depending on that permeability, if permeability is really high, it most likely will reach the endpoints of the model. So basically that's how it works. I have an initiator source, and that source also has a parameter called sigma s, which is the source flux, and depending on how I vary that parameter, that firing of that source would be stronger, would be weaker, and that way I can also see the reach of the calcium on the different cells related to plasmodesmata. and Siefken.
What tools or programming languages are you using to develop and test the model?
I'm using Python, so all of that code is being performed in Python, and I'm using VS Code as my application, as well as Google Colab to work with my mentor. Google Colab is how my mentor teaches me stuff, because Google Colab is really easy to understand. But to actually do the proper applications, I need a more robust app and programs such as VS Code. That's what I use in most cases and in most simulations. That's all Python.
So in your biomedical engineering major, you have to learn about Python, or it's something that you had it before you coming to Purdue?
When I came to Purdue, I had absolutely no knowledge about coding.
Okay.
Then my first year, I learned MATLAB in my engineering courses. Then in my first year, my second semester, I learned C, it's another language. But I didn't know much Python until I was actually in biomedical engineering, and I had courses that require some Python knowledge. So some courses taught us Python, but for research itself, I needed to learn a lot from myself, because the codes and programs I'm using are way more advanced than I was used in class. So I've got a lot of help from my mentors, and my PI, Dr. Piner. She's taught me a lot on how to code, and basically the Python that I've learned it a little bit through classes, but mostly once I got into the lab itself.
Okay. Can you walk me through a typical day working on this research?
So it's coding. So I don't go to a physical lab. It's not a physical facility where I go and I do experiments. I use my computer. I can do it from my house, from the BM building, in the library, any place. And it can be tedious sometimes, and it can be frustrating. You just look at a block of code and changing parameters, writing code, and maybe the code doesn't work, you get damaged, so you gotta troubleshoot it. You gotta spend half an hour, 1 hour looking through code to see that your mistake was probably only a comma or a semicolon, which can be frustrating. But when you get results and when you see actual graphs and your code actually runs and works, it's really, really satisfying. And it's really satisfactory to see that all your work is yielding results at the end. Even though that process can be frustrating and can be really long, then results are always really satisfactory. But yeah, it's my days when I do research, usually open my computer, start writing code, and stay writing code 1, 2, 3 hours, troubleshooting, getting results. Yeah.
Nice. It's interesting because Kenneth, he's also my friend, and I've never known that he already he was a research student. And for me, it's very interesting to discover this, and also about his knowledge in this kind of research project. That for me, it's something new. I'm a financial planning student, and you are a biomedical engineering, but I didn't know that you were focused on that kind of research project. Kenneth, what challenges have you faced while building or validating the model so far?
I faced a lot of knowledge challenges, because sometimes to be able to apply new agents or new species, code is required that's way beyond my level of knowledge. So I've gotten stuck several times. As well, I've gotten stuck in plotting functions as well, because graphs just don't work and I have no idea what's going on there. So to target those problems, I usually reach out to my mentor, which is a PhD student, so he knows way more than I do. Or I also reach out to Dr. Pina, that's the professor, that she also knows a lot, way more than I do. And when I reach out to them, they always are able to help me. But the main problems I encounter are basically knowledge gaps or bugs or errors that I do while performing code that I don't notice or that I have no idea how to solve. So it's basically those knowledge gaps that I usually have to overcome, either by learning, reading literature, or reaching out to my mentors.
How do you verify whether the simulations reflect real biological behavior? I mean, how you observed any unexpected or surprising results in your simulations.
So first, there's a lot of literature involved. I have to read a lot of scientific papers, manuscripts, to understand more or less what I should see. And then after I run a code and I see something completely ridiculous, I know that that's not what should be going on and I have to go back. But yeah, I usually try to compare my results to literature. Of course, what I'm researching is new, so I'm not gonna find everything, but I can sort of guide myself to see that what I'm getting makes sense and it's biologically possible, and it's not biologically impossible because there's some stuff I've gotten that I say, no, this cannot happen, it's impossible. So it's basically understanding that type of plant you're using, the type of soil you're using, and depending on your parameters to see that if what you're seeing would make sense, it is possible, or it's just absolutely impossible. So it's basically just learning, understanding, reading manuscripts, and comparing some graphs in previous researchers have made to the new ones we're creating, and see if they have some correlations, make some sense.
So you said the results depends on the types of plants are you using.
Types of plants and the changing parameters.
And the changing parameters. What kind of plants did you use in this kind of model?
Right now I'm using Arabidopsis thaliana. Those plants have a cell that have cotyledon pavement cells. Those are the ones that I'm using right now.
Okay.
7-day-old plants as well.
7 days.
7-day-old, yeah.
Oh.
So it's baby plants.
Oh wow, interesting. Kenneth, what are the potential real-world applications of this research in agriculture or biotechnology?
So in agriculture, it's really important because there are pathogens in the wild, and plants communicate those threats using calcium signaling. So if you're able to make computational models that we're using to simulate those pathogens and simulate those stimulus, those responses, we would be able to create applications in the future by understanding how those pathways work and perhaps creating some sort of artificial signals, artificial stimulus that would make the plant yield some reactions that we would like to have them artificially for different purposes depending on the agriculture's goals. As well, it would allow us to make potentially plants immune to certain pathogens by making them respond before the pathogens are there, making them essentially immune. As well, there are plants that have immunity to some pathogens, and there are plants that have not the same immunity to the same pathogens. But if you're able to understand how, for example, plant A attacks those pathogens through calcium, we can then artificially simulate those same responses in plant B that doesn't have those sorts of instincts or knowledge or DNA. And that way we can make plant B immune as well artificially, helping agriculture a lot, helping crops, and saving plants overall at the end.
Oh wow. Kenneth, how has this research influenced your academic or career goals?
So, I said before, I love biomedical engineering. I love the bridge between biology and math. Now, I don't want to stick only with a bachelor's degree. I want to move forward. I want to do a master's. So, I'm gonna do a 4+1 program that the BME school offers. So, thanks to this lab, I will be able to do a thesis in my master's because I'm gonna stay in this lab through my master's. So this is really beneficial. It's a great opportunity because I'm able to get a thesis out of my master's thanks to this lab. So it has really influenced me in the fact that I don't only want to stay with classes and bachelor's degree. I want to move forward, do research, and do a master's degree, do a thesis in this lab.
Okay, so do you have plans to continue working on this? And do you want to get a master's degree after your undergrad, right?
Yes, exactly.
Oh yeah, and what is the master's about? Right?
The master's is basically I take more 500 level courses, and as well, since I'm gonna do the thesis master's, not a professional master's, I need a lot of research credits as well. So one research credit is around 5 hours of research per week. Last semester I did 2 research hours, so that's 10 hours of research per week. This semester I'm doing 3, that's 15 hours. In the master's degree, I'll most likely do 4 credits per semester, so like around 20 hours of research per week. And I will need to craft a thesis in this project, so it's gonna be still be about classroom signaling most likely. And basically I would need to do way more research than I'm doing right now. I'm gonna get a lot more knowledge and get a thesis out of it, and that way get my master's degree with a thesis. Thanks to this lab.
Okay. Kenneth, I remember you mentioned me that you want to apply to do a semester abroad next semester in Madrid, right? Tell me a little bit about this experience and how you can manage this kind of project there. Like if your experience there could you can involve your experience there in this kind of project, or are you gonna just focusing on like taking some classes of your major, or what are your plans on this project?
So next semester I'm studying abroad in Madrid. So I'm studying Carlos III. That's the university with which the biomedical department has like a contract. So I will be taking classes there that will transfer into Purdue credits. So I wouldn't get behind. I would be able to take classes of my major while being there. As well, I talked with Dr. Pinar about continuing in the lab while being abroad. So thanks to the fact that my lab is not like a physical place I go to, I can just take my computer anywhere in the world and code. And the only thing that would be challenging would be the meetings because of the time difference, but that has happened before in the lab. There's people in the lab that have been in Greece, have been in Ireland, and just connect at different hours at one specific time. But they are still able to be in this lab and get results and still be able to work. So that's something really beneficial of the fact that my lab is computational. So I will be able to continue my project and my work while being abroad. So I would still be able to do what I love and get more results and progress towards my degree while being in my lab in Madrid.
Wow, it's interesting how technology, works different right now, and you can work in whatever place you want, and you are staying. Kenneth, how do you see yourself in 10 years?
So in 10 years from now, I'll be 30. It depends. So I'm thinking after my master's degree, well, I graduated my undergrad with 21 years, then my master's will be 1 more year, so I will graduate with 22 years. I would like to do an MBA, it will be 2 more years, that will take me to 24 years. Then I would like to work in industry. I would like to work in a probably lab for some time, and then get into research probably in the future. I would like to do a PhD, probably not immediately after I finish my MBA or anything, but after some time working in industry. That way I can get a PhD and then probably work in a world-renowned lab, probably in Switzerland or here in the US or in Canada and Europe.
So are you planning to do the MBA here in Purdue or other place?
My master's, I'm gonna do it here at Purdue, but my MBA, I would like to maybe do it on another place. I would like to probably explore more. If I end up really liking my semester abroad in Madrid in Europe, I would like to do my MBA in Europe, maybe in Germany or another country, just to get a more global experience. That way, I'm not sticking only with the US education, but also getting education in Europe, some European diplomas as well.
Kenneth, how many languages do you speak? I'm sorry for the question, but
Fluently, I speak English, Spanish, and French,
wow,
and I speak a little bit of German.
very nice. Okay. What kind of advice would you give to students who want to start doing scientific research as undergrads?
First, don't get desperate. When I started to look for a lab, I got really desperate because I would email a lot of professors and I wouldn't get responses a lot of times, or sometimes I got interviews, but anything ended up coming out of them. So it can be frustrating at the beginning, but you just need to be persistent. Don't be afraid, don't be shy to walk up to your professor, even if it's at the end of class or just knocking at their door in their office and just talking to them. If you're not shy and you're persistent, you will eventually get a lab that you're interested with and you like. At first you might get reactions and professors telling you there's no space or just not responding to emails, but just don't get desperate. You are gonna be able to get something that you like and you're gonna end up being really well. As well, once you are in the lab, it's gonna be very scary. Remember my first lab meetings, were a bunch of PhDs giving presentations and talking about stuff I had no idea about. It was like Chinese to me. And I was like, wow, I'm that wrong here, I don't know anything. But no, like, they are there to help you. So as long as you put in the effort and you're not afraid to reach out and ask for help when you need, you are gonna get places and you're gonna be satisfied with your work and you're gonna get results and everything's gonna be okay.
Well, thank you very much, Kenneth, for your time and for sharing all of this knowledge that you are doing on the last recent months on this project. See you soon. Thank you.